DNA is a very pretty molecule. It's the DNA double helix. But in order to understand its structure, which we'll study in great detail later, let's just look at a single strand of DNA and examine how the polymer is put together. So that's what we'll do right now. And I'd just like to point out that I'm going to spend a little bit of time on this because this is such an important molecule. This is the hereditary material, and I really would like you to understand its structure in some detail. We'll need to know its structure when we want to understand DNA replication and other processes later on. So we'll spend a little bit of time on this now. So what we have here is a short stretch of single-stranded DNA, not double-stranded DNA, not double helical DNA. And we can see that it is made out of component nucleotides, and each nucleotide in this 4-nucleotide stretch here, we're only showing a 4-nucleotide stretch as an example. Each nucleotide has a nitrogenous base, having nitrogens shown in blue. Hydrogens are not represented here for simplicity's sake. And then the second component is a ribose sugar. There's a ribose sugar. Carbon atoms are shown in gray, and oxygens are in red. And you can see that an oxygen lies at the apex of the sugar. And then the third component of every nucleotide is a phosphate group. Phosphorus plus oxygens. And those are the three components of every nucleotide in a DNA polymer. So the nucleotides are the monomers, and DNA is the polymer. So that now we can consider how those are the linkages that connect the various atoms of the molecule. So considering the ribose sugar, there's an oxygen at the apex of the ribose sugar. There is the first carbon of the sugar ring, second, third, fourth, and fifth carbon. And now let's indicate that there is a polarity to the strand because the linkages go from the 5' carbon to the 3' carbon and then on down the molecule. So there is a polarity to the chain dictated by the polarity of the sugars and the phosphates. So if we look at our phosphorus atoms now and consider what is attached to them, and this molecule could go on up and down for long stretches. We're only showing a 4-nucleotide stretch. But naturally, because we're talking about phosphate groups, there are oxygens attached to the phosphorus atoms in the phosphate functional groups. And those oxygens can have hydrogens on them that they donate to solution. And that makes for an acid. When hydrogen ion concentrations go up, we have an acid, which is donated hydrogens. Now if we look at the sugar and look at the second carbon, we see that it does not have an oxygen attached to it. So it is deoxy, minus a hydroxyl group. There's no oxygen attached, no OH group attached to the second carbon. In RNA there would be, but this is DNA. So we have deoxy ribose sugars. And since we do have ribose sugars as a main component of every nucleotide, then we say that there's deoxyribo, standing for the ribose. And we know that the nucleic acids are so named because they were first discovered in the nucleus. Even though RNA is found in the cytoplasm as well, nevertheless, RNA and DNA are known as nucleic acids because DNA was discovered in the nucleus. So we have deoxyribonucleic, and then the oxygens on the phosphates, remember, can donate hydrogens to solution, making the phosphate groups, the phosphate functional groups, acidic because they increase the hydrogen ion concentration in an aqueous solution. Therefore, we have an acid, and DNA is an acid. So DNA, deoxyribonucleic acid. Now, considering showing the individual monomers now in color, we're showing a nucleotide in blue that has A as its nitrogenous base, adenine. The yellow nucleotide has thymine as its nitrogenous base, red cytosine, and green guanine as its nitrogenous base. So you can see the individual monomers clearly here. And if we zoom in on the nucleotide containing cytosine as its nitrogenous base and number the carbons in the sugar ring, 1, 2, 3, 4, 5, it's carbon 1, and the nitrogen of the nitrogenous base is linked to the first carbon of the sugar, and that is called a glycosidic bond. And it's always a nitrogen of the nitrogenous base that is linked to the first carbon of the ribose sugar. And glyco implies sugar, so a glycosidic bond links the nitrogenous base to the ribose sugar. Now, if we consider how the sugar is linked to the phosphate, it's linked through an oxygen to the phosphorus atom, and that phosphorus atom is linked to the next oxygen that is tied, which is covalently bound to the third carbon of the nucleotide above it. And so oxygen linkages are called ester linkages, and we therefore have a phosphate with 2 ester linkages on either side of it. I should say a phosphorus atom with 2 ester linkages on either side of it. And you can see that if you remove the phosphates, those are the linking points for the polymer. So we have a phosphodiester backbone, phosphodiester linkage. There's the phosphate, and there's one ester linkage, and there's the other one. So we have a phosphodiester linkage that connects the backbone, connects the monomers in the backbone. So that we can then consider the molecule as a sugar phosphate backbone, here shown in purple, sugars and phosphates. Sugars and phosphates. And that's the backbone. And then the nitrogenous bases hang off the sugars, and in double helical DNA they are available for hydrogen bonding to bases from the other strand of DNA in a double helix. Note that the nitrogenous bases are not connected to each other except, in covalently speaking, they're not connected to each other except through the phosphate, the sugar phosphate backbone. All right then, having done that, it now makes it easy to understand the schematic representations that you often see. So here is a short stretch of single-stranded DNA shown schematically with the numbered carbons of the sugar rings, the nitrogenous bases, here's the glycosidic bond, and the phosphates connecting the sugars through a diester linkage. The oxygens aren't shown here. Notice that we are deoxy on the 2' carbon, no oxygen there. And these are the nitrogenous bases. This uracil is only found in RNA, not DNA. DNA has 4 nitrogenous bases, 2 purines, adenine and guanine. These are double-ringed aromatic rings. Aromatic ring means that they're carbon rings mostly with double bonds. And these electrons in these double bonds can resonate around the ring. They're called aromatics. So we have 2 purines with the double aromatic ring and 2 pyrimidines, cytosine and thymine, smaller single aromatic rings. And those are in DNA, G, T, C, or A is possible at any given position along a long stretch of DNA. We'll talk about the genetic code later. So we know that DNA encodes amino acids, it encodes proteins, and DNA exists in a double helix. And there are base pairing rules. So we have one strand of DNA here, another strand here. The nitrogenous bases are kind of like are in the center of the molecule along the long axis, and they are arranged. The 2 strands of DNA that wrap around each other are arranged such that hydrogen bonds form between adenine and thymine and between guanine and cytosine. Here's adenine and thymine. But only between those base pairs. Adenine can't pair with guanine or cytosine. Cytosine can't pair with adenine or thymine. T's are always paired with A's, and G's are always paired with C's. Here's an AT base pair there. So we'll talk more about that a little later in the course. RNA is similar to DNA, but it contains ribose instead of deoxyribose, so there is a hydroxyl group on the 2' carbon, the second carbon of the ribose sugar. And RNA contains uracil instead of thymine. And RNA is usually a single strand, not double-stranded, although some double-stranded RNAs do exist. And RNA is the carrier of information from DNA to specify the manufacture of a particular sequence of amino acids in proteins. Again, a process we'll study later. So here is DNA and RNA compared. Double helical DNA, thymine, whereas RNA has uracil instead of thymine. Single-stranded RNA, double-stranded DNA, hydrogen-bonded base pairs in the interior. And, of course, DNA, the 2' carbon is deoxy, whereas the 2' carbon in RNA has a hydroxyl group on it, not shown here. There are other nucleotides than the ones found in DNA. For example, ATP, shown here, adenosine rather, which means there is an adenine nitrogenous base, and the sine implies that there's a sugar attached to that nitrogenous base. So here we have adenosine, adenine plus the sugar, ribose sugar. Note that it is not deoxy, and 3 phosphates. So this stands for, ATP stands for adenine here, adenosine rather. Adenosine, that's these moieties together, these two. Triphosphate, 3 phosphates, adenosine triphosphate. And this is the primary energy currency of the cell, because these breakage off of phosphates here releases energy that can be used and can be harnessed by enzymes to catalyze biochemical reactions. So ATP is the energy currency of the cell. And there are also NAD and FAD that are electron carriers. And we will see that the passage of electrons in redox reactions. Remember, we talked about reduction oxidation. So NAD can be reduced to, it can accept basically an electron on a hydrogen, and it can become NADH and become reduced NAD by accepting an electron. And FAD can accept an H and become reduced FAD. And then that electron can be passed on to other molecules, and in so doing, that can release energy that can be harnessed for various purposes, as we will see. So that's something to keep in mind. We'll see that again. Now we're ready to tackle proteins, and so we'll go on to the next part for that.